create an artificial cell that grows and divides like a natural one. It is a remarkable milestone for genetic engineering that scientists have developed a single-cell synthetic organism that grows and divides much like a normal cell. This newly developed cell is known as JCVI Syn3A. While the work's roots can be traced back to the 1990s, the most recent strides occurred this century, with researchers in 2003 successfully synthesizing a small virus that infects bacteria. That led to a new breakthrough in 2010, with scientists at the J. Craig Venter Institute, JCVI, in Maryland designing the first synthetic bacterial cell, called JCVI Syn 1.0. JCVI Syn 1.0 is the first organism on Earth with an entirely synthetic genome. It is engineered by stripping the natural DNA out of the bacterium Mycoplasma mycoides. That means they destroyed the DNA in those cells and replaced it with DNA that was designed on a computer and synthesized in a lab. In 2016, researchers had then created synthetic minimal cells, called JCVI Syn 3.0, which only possessed 473 genes in total, shorter than any known self-sustaining, living organism in the natural world. They were able to grow and divide on agar to produce clusters of cells called colonies. But on closer inspection of the dividing cells at the time, Venter and his colleagues noticed that they were producing daughter cells of bizarre shapes and sizes. Scientists took on the laborious task of constructing dozens of mutants in which genes, both as individual genes and in groups, were added back to JCVI Syn 3.0. Now, J. Craig Venter Institute, JCVI, in collaboration with the National Institute of Standards and Technology, NIST, and the Massachusetts Institute of Technology, MIT, Center for Bits and Atoms have figured out a way to prevent these strange morphologies from occurring. With a newly modified variant of JCVI Syn 3.0, known as JCVI Syn 3A. JCVI Syn 3A features 19 new genes. Only 7 genes are thought to enable the artificial cell to reproduce in a more regular manner. The researchers and scientists have published their findings in scientific journal Cell.